What is up, YouTube? Back here today with a pretty awesome MCAT question. You'll see today that even though this question may not seem the most difficult, it's a great way to understand interesting concepts and also understand relevance to disease. So it says the Nobel Prize in Physiology and Medicine in 2017 went to Yoshinori Osumi for his discoveries of the mechanism of autophagy. Autophagy is a process through which a cell degrades and recycles its own cellular content. What organelle is most closely associated with autophagy? And then you have 5 answer choices. You may know the answer right off the bat, but that's not what I'm trying to get you to see in this video. I'm trying to show you something much more relevant here. I want to try to show you how you can think about these mechanisms. All right, so what is autophagy? Autophagy is basically this crazy mechanism shown right here. All right, and I'm going to use orange now, where you have waste in the cell that eventually gets broken down. So if you look at the last step of this, you'll see that the waste is broken down. Basically, what actually ends up happening is all this stuff here is waste, cellular waste. All right. There's a lot of stuff. Your cell is like a person. It creates waste. And what happens in the cell is basically around that waste, a vesicle membrane will form. And the mechanism through which the vesicle membrane will form is right here. And the mechanism through which it forms is actually unknown. So think about how crazy that is. Even though we know what autophagy is, we still don't really understand how it works. But let me just say that what ends up happening is once we have all our waste in one place, it's kind of like the garbage collector of our cell. Then we're going to mix it up with these hydrolase enzymes. Okay, these are hydrolase enzymes. Hydrolase enzymes are known to hydrolase. They put water in to break down bonds. And what ends up happening is that you end up getting protein slash waste breakdown because you have your waste here, and all of that waste is going to be broken down. Okay, broken down. All right. So that's the process of autophagy. More importantly, how does this relate to disease? It's always important to see how these simple questions, are not just stupid, are not just there to trick you. They're intended to show you there is some sort of relevance here. All right. In this case, think about neurodegenerative diseases. Okay. This is just one example. So you'll think about Alzheimer's disease, and in this case, what you'll see in Alzheimer's disease is people with Alzheimer's. After death, when you do the autopsy, you'll see that their brains have an increase in the amount of beta amyloid. That's a protein, okay? Beta amyloid is a protein. Another thing that's a protein is tau protein. Both of those proteins are present in higher amounts in the brains of Alzheimer's patients compared to normal patients. And what might be happening here? Well, if these proteins are building up to excessive levels, it's clearly that the cell does not know they're building up to excessive levels. Like it doesn't have a mechanism to keep them in check. So in this case, the autophagy here is downregulated. And you'll see that the autophagy is a critical component in the development of Alzheimer's. Now, let me just give you the flow here. If you look at the upper right corner, you'll see that no autophagy means that waste builds up. And if waste builds up, that can be even worse because you get damage to DNA then, right? Think about your own house. What if no one ever took out the trash? It would be a crazy place, right? Same thing with the cell. If autophagy is downregulated, much like it is in the disease of Alzheimer's, you could get buildup of proteins that could lead to disease. And I'm not gonna say that these proteins are causal for Alzheimer's. They're just associated with Alzheimer's. So with that being said, let's investigate our initial answers, right? We had mitochondria, ribosomes, lysosomes, rough ER, and none of the above. And again, this is not a hard question. I'm sure some of you knew that the answer here was what it's going to be, but let me just go through these. So you should know for every organelle some of the keywords that it should be associated with. For example, for mitochondria, you should know that it's linked to the citric acid cycle and the electron transport chain, which are part of respiration. You should also know that it's linked to beta oxidation, which is used for fat breakdown. I've mentioned this a lot. These are the key things you should know that mitochondria do. More importantly, you should also know that mitochondria are only in eukaryotes. All right. So knowing what kind of cells have what organelles is important. Now, ribosomes. There's about 10 million per cell. So you have 10 to the 10 million per cell if you were in a million cell. The ribosome is actually present in all cells, right? Because ribosomes, believe it or not, ribosomes, I mean, I'm sure you do believe it because it's something you've heard since third grade. I don't know why you wouldn't. But they help translate proteins, and all cells need proteins to live, okay? And that's why ribosomes are present in all cells, because all cells need proteins. But the ribosome itself is made of rRNA and protein. So isn't it crazy that a protein is helping make more proteins? It's a circular kind of dilemma here, where you have a protein that makes more proteins. It's inceptionated. More importantly, again, these are the general things that I would associate with ribosomes when I think about the top 5 things about ribosomes off the bat. These are definitely the 5 of the things that I would remember. Now, lysosome. Again, lysosome is a membrane found mostly in eukaryotes, right? The other thing is that the pH in a lysosome is about 5. Why is it 5? Well, it's 5 because it's used in the breakdown of things, right? The things that we use to break down other things are lysosomes. And the pH of 5 helps these hydrolase enzymes work efficiently. And I guess at this point, you should know that I just gave away the answer because, yes, lysosomes are the organelles associated with autophagy because they break down a lot of the waste in the cell and they have a lot of hydrolase enzymes. And so if we go back to our previous picture, you'll see that I was referring to this thing right here. You'll see I'm highlighting again in orange. I'll maybe make it a bit thicker. Oops, sorry. Let me make it thicker. You'll see that this part that I was referring to, this is a lysosome. So the lysosome fuses with the waste you have, and it actually causes the degradation. So the answer here is clearly C, okay? But just because I want to make sure we all understand certain things, let's go to number D. It's the rough endoplasmic reticulum. So again, this is an organelle in eukaryotic cells. That's something you should pick up. And it's also, you should know that it has ribosomes on its surface. That's why it's rough. All right, and if it has ribosomes, you probably can assume it's linked to making proteins, and it is. It actually develops and traffics proteins. It actually develops and modifies proteins that are leaving the cell. So ribosomes in the cytoplasm make proteins that will end up in the cell, but ribosomes on the rough ER help make proteins that leave the cell or end up in the cell membrane. So with that being said, I already told you the answer here was C. And this is not a hard question, but I want you to see how it can be a hard question, because this question is literally the same as this question below. But this question below is much more related to medicine. All right, so I wanted students to see that even though something sounds like a very factually based question, it actually has a lot of scientific underpinnings. And you'll see the question below. It says, in Alzheimer's disease, a protein called beta amyloid builds up in and around neurons. The inability to break down excess amounts of beta amyloid protein is due to pathophysiology in which organelle? Now compare that to the first question. The second question is clearly way more difficult, even though it asks the same thing. And that's the point I wanted to make here. Just because a question sounds basic doesn't mean it has to be. I could have easily asked you the second question, and you would have maybe been lost compared to the first question where I kind of broke it down. So that's why you always want to think about the clinical relevance of a question. So with that, the answer is C. Hope you guys enjoyed the video. I'll be making a lot more soon, and I'll see you in the next one. Thanks for watching.
Hey, thanks for watching the video. Really appreciate it. You want to check out any of my other videos, there's going to be one right here. Another link to one of my videos right here. And another video right here. Why not? I'll put one video right over here. And last but not least, if you want to subscribe to this channel, really appreciate it because I'm still an early YouTuber trying to get it down. But a subscription button should be right over here. So please subscribe. Cool. Thanks. See you guys in the next one. Hope you find these videos helpful.